Hello, greetings. Thank you for joining. Welcome to our RRP hosted research webinar series, where we really aim to bring you the latest and greatest RUNX1 research updates from the leaders in our RUNX1 field. I'm Katrine Erickson. I'm the Executive Director of the RUNX1 Research Program. There may be some of you who don't know RRP, so to introduce you to who we are a nonprofit organization dedicated to supporting patients with RUNX1 familial platelet disorder, a condition driven by germline RUNX1 mutations that we now know can lead to many different health issues. But of course, the most concerning issue is the development of hematologic malignancies, and predominantly those are represented by AML and MDS. RRP is laser focused on one goal, and that is to prevent cancer and improve the quality of life of RUNX1 FPD. We do this through funding research, through building and supporting our patient community, and through facilitating collaboration across all stakeholders within our healthcare ecosystem. And we're doing this for that one goal, which is to discover treatment interventions, again, that will prevent the development of hematologic malignancies in our patient community. I would like to also, of course, acknowledge all of our funding partners. Without their partnership, we would not be able to have committed as much as we have since inception. We're up at over $11 million in RUNX1 FPD research funding. So our co-funding partners include Alex's Lemonade Stand Foundation, the Leukemia Lymphoma Society, the Mark Foundation for Cancer Research, Edward P. Evans Foundation, and the National Cancer Institute, as well as NIAID. If you're conducting RUNX1 FPD research or RUNX1 relevant research and are interested in presenting your research findings at one of our research webinars, please do reach out. We'd love to hear from you and give you that opportunity. Also, if you're interested in applying to any of our grant programs, please keep checking our website and follow us on social media. We don't have consistent cadence of our grant programs, but we do continue to fund research, as I mentioned earlier. So please just keep checking in with us. Okay, so before I introduce our renowned speaker today, I just wanted to give you all a quick reminder that we'll host a Q&A session at the end of the presentation for about 10 to 15 minutes. And so the best way to ask your questions is to use that Q&A icon. Submit your questions through that, and then we can make sure to address those questions as best as possible. If you have any comments, of course, you're more than welcome to put those into the chat icon function. Okay, it is with great pleasure that I introduce Dr. Nancy Speck, who is the chair and John W. Ekman Professor of Cell and Developmental Biology at the Perelman School of Medicine at the University of Pennsylvania. She is also the RRP chair of our scientific advisory board and has been an incredible partner and leader and advisor to our program. Dr. Speck has devoted her career to understanding the role of RUNX1 in establishing the hematopoietic system and how inherited RUNX1 mutations predispose to leukemia. I don't think it goes without saying, but I'm going to say it anyways. She is world-renowned for her RUNX1 research, and she continues to make significant discoveries that deepen our understanding of this critical hematopoietic transcription factor. So the work she will describe today is entitled RUNX1 Attenuates Toll-like Receptor and Type 1 Interferon Signaling in Neutrophils. And this work was partly funded by RRP. So Nancy, we're delighted to have you here with us, and I am looking forward to your presentation, as I know many of our attendees are as well.
Thank you, Katrine, for the really kind introduction, and I'm looking forward to sharing the work with you all. Let's see. Is that good?
Yep, all set.
All right, so I'm really actually very excited to share the story with you. I really believe it begins to get at the crux of what could be an important mechanism by which inflammation and leukemia risk are increased in patients with RUNX1 mutations. So, as probably all of you know, RUNX1 is a DNA binding subunit in the small family of core binding factors, and it can function as either an activator or a repressor of transcription based on the partners it's using and the chromatin modifying complexes with which it associates. Mutations in RUNX1 and in the gene encoding its non-DNA binding partner, CBF-beta, are somatically mutated in multiple types of leukemia listed here. And inherited RUNX1 mutations cause familial platelet disorder with predisposition for myeloid malignancy, or FPDMM, which of course is the topic of my talk today. So FPDMM is a rare disease that's caused by RUNX1 haploinsufficiency. Well-established symptoms of FPDMM include low platelet counts and platelet activation defects, and an approximate 40% lifetime risk of leukemia. In addition, FPDMM patients have accelerated clonal hematopoiesis, which most likely is contributing to this increased leukemia risk. So clonal hematopoiesis refers to a state in which a disproportionate fraction of blood cells is derived from a single clone of hematopoietic stem cells, or HSCs. And it's defined clinically as greater than or equal to 4% of blood cells being produced from a single expanded HSC clone. Clonal hematopoiesis is caused by the acquisition of a somatic mutation, generally in a leukemia driver gene, and this results in an expansion of that HSC relative to other HSCs in the bone marrow. And the phenomenon is called clonal hematopoiesis of indeterminate potential, or CHIP, when there's no evidence of a blood abnormality. So the blood cells themselves look phenotypically normal. However, CHIP mutations can increase the inflammatory properties of myeloid and lymphoid cells that differentiate from the mutated HSC. And then inflammatory mediators produced by these mutated immune cells can in turn selectively impair normal HSCs, while the mutated HSCs have acquired mechanisms that allow them to persist despite this inflammation. And so this provides a selective advantage to the mutated HSCs, which allows them to further clonally expand. So you can see there's a pernicious cycle that ensues in which the mutated HSC clone can increase in size, give rise to even a greater proportion of mutated inflammatory cells, which then can act back on the mutated HSC and normal HSCs. Now, the presence of CHIP does elevate the risk of several diseases. So, for example, if the mutated HSC acquires additional somatic mutations, CHIP can evolve into leukemia. And although the overall leukemia risk is still relatively low, it's still about 10-fold higher than in individuals without CHIP. In addition, increased inflammation from the mutated immune cells increases the risk of inflammatory disorders, such as cardiovascular disease. Now, the incidence of CHIP in the general population increases with age, and by age 70, approximately 15% of us will have CHIP. But in FPDMM, the rate of CHIP is greatly accelerated and its penetrance is much higher, and this correlates with the elevated risk of leukemia. So this brings us to what I think is a central question in FPDMM, and that is, what is driving this more penetrant, highly accelerated rate of CHIP? And we hypothesize that a baseline increase in inflammation in patients is involved. So there's several lines of evidence that inflammation is elevated in FPDMM. So first, about 10 years ago, Ravi Bhatia described an FPDMM family with 14 family members in which all 7 mutation carriers had mild to severe eczema, which is an inflammatory skin disease. And more recently, the National Institutes of Health has launched a longitudinal natural history study of FPDMM, led by Paul Yu, which has enrolled over 100 patients since it opened in 2019. And the study protocol involves, amongst other things, a detailed clinical evaluation of patients. And one of the findings is that patients have an increased incidence of allergic and inflammatory symptoms compared to the rest of us, including asthma, obstructive sleep apnea, eczema, allergic rhinitis, and recurrent infections. Furthermore, Anapriya Agarwal at Oregon Health Sciences University, in collaboration with Paul Liu in the natural history study, showed that the bone marrow supernatant from FPD-MM patients contains increased levels of inflammatory cytokines and chemokines. And so we reasoned that an inflammatory bone marrow could provide the conditions that select for the expansion of FPD-MM HSCs that have acquired CHIP mutations. So the story I'm going to present today is about a mechanism by which RUNX1 loss increases inflammation, which I think is very important to understand if we hope to control it. And the majority of the work was done by Alex Zazulin, an MD-PH student in the lab, in very close collaboration with Wenbao Yu, a bioinformatician at CHOP, and the work was funded by the RUNX1 research program. So the story begins with a mouse model in which we deleted RUNX1 in HSCs, and then we analyzed inflammatory cytokine production by bone marrow cells to identify which inflammatory cytokines were secreted in excess and the cellular source of those cytokines. So deletion of RUNX1 in hematopoietic stem cells results in a mouse with very few lymphocytes and very impaired megakaryocyte development, because RUNX1 is very important in the development of these lineages. So therefore, we focused our attention on inflammatory cytokine production by myeloid, RUNX1 deficient myeloid cells. Now, we first analyzed the response of myeloid cells to toll-like receptor ligands. So toll-like receptors bind pathogen-associated molecular patterns. So for example, toll-like receptor 4 binds to lipopolysaccharide, which is an outer membrane component of gram-negative bacteria. And we suspected TLR4 signaling might be affected by RUNX1 loss, because over the many years that we've studied and cared for these mice, we've observed changes in their overall viability depending on where they were housed, including in different rooms in a single mouse facility. So for example, we once made and studied a RUNX1 mutant mouse line at Dartmouth when I worked there, and which Mark Reimakers has been working on at Erasmus University, but we cannot generate live animals at Penn, which is apparently has a less well-maintained facility. And so because of these differences in viability, we suspected that this could be caused by differences in the levels in the microbiomes or in levels of bacterial pathogens in various rooms or facilities that could be activating toll-like receptor signaling. So we analyzed cytokine production by bone marrow cells, or in this case, purified neutrophils and monocytes from the bone marrow, and stimulated them ex vivo with the TLR4 ligand LPS, and then measured the amount of inflammatory cytokines in the supernatant 8 hours later using a cytometric bead array. And when we found that when we deleted RUNX1 in all hematopoietic cells, inflammatory cytokine production by bone marrow cells increased, and that neutrophils in particular produce copious amounts of a handful of inflammatory cytokines and chemokines, including tumor necrosis factor and CCL3. And in fact, RUNX1 deficient neutrophils produced as much TNF as monocytes on a per cell basis. And since neutrophils represent 40 to 60% of hematopoietic cells in the bone marrow, we reasoned that they may be an important source of these excess inflammatory cytokines. There was a trend towards an increased production of inflammatory cytokines by monocytes, but for the most of TNF in particular, but for the most part, RUNX1 deficient monocytes did not overproduce inflammatory cytokines. We also measured cytokine production by neutrophils purified with mice with a monoallelic loss of function RUNX1 mutation that we obtained from Lucio Castilla. This is a more faithful model of FPD-MM, and we found small but significant increase in the production of TNF and CCL3. However, since the phenotype is much more robust when both RUNX1 alleles are deleted, we used mice with biallelic RUNX1 deletions for all of our mechanistic studies. So when we delete RUNX1 and HSCs with VAV1-CRE, that depletes it from all downstream blood cells. And as I mentioned, pan-hematopoietic RUNX1 loss affects several blood lineages, including megakaryocytes and lymphocytes. So this raised the question of whether the increased inflammatory cytokine production by neutrophils was intrinsic to the neutrophil lineage or whether perturbations in other hematopoietic cells, so for example, the loss of Treg cells, was indirectly responsible for the neutrophil phenotype. So we address this question by generating mice in which neutrophils are raised in a largely normal bone marrow. So what Alex did was to transplant mice with a 10-to-1 ratio of wild type versus RUNX1 mutant bone marrow cells to create recipient mice in which the wild type bone marrow cells far outnumbered the RUNX1 mutant cells. And this graph shows the excess of bone marrow cells derived from the wild type donor versus these RUNX1 deficient cells in red in the transplant recipient. She then purified the wild type and the mutant RUNX1 mutant neutrophils from the transplanted mice, stimulated them ex vivo with LPS and measured cytokine levels. And what she found was that the RUNX1 deficient mutant neutrophils still overproduced inflammatory cytokines, demonstrating that this hyperinflammation was an intrinsic property of the neutrophils and was not caused by RUNX1 loss in other lineages. So as I just showed you, if we knocked out RUNX1 in HSCs, the neutrophils were hyperinflammatory. However, when we knocked out RUNX1 in neutrophils using a different Cre transgene, they were not inflammatory. And so this demonstrated that RUNX1 is not acting in neutrophils per se to restrain inflammatory cytokine production. Rather, it suggested that RUNX1 is required in a neutrophil precursor, perhaps in the hematopoietic stem cell itself or in some downstream progenitor to restrain inflammatory cytokine production by neutrophils. Single-cell RNA-seq indicated that an inflammatory signature was first evident in the granulocyte monocyte progenitor, or GMP. So therefore, we knocked out RUNX1 in GMPs using CEBP alpha Cre, and this resulted in inflammatory neutrophils. So these results suggested this was an epigenetic phenomena, and the mechanism involved chromatin changes which were occurring in GMPs when RUNX1 was lost, and those chromatin changes were being subsequently transmitted to the neutrophils that differentiate from them. So to gain insights into this epigenetic mechanism, we examined the chromatin accessibility of GMPs and neutrophils from control mice and from mice in which RUNX1 was deleted in GMPs, and these are referred to as RUNX1 delta GMP mice in my slides. And we performed this analysis in the absence of LPS to examine the chromatin changes at baseline. So we first examined global chromatin changes caused by RUNX1 loss in GMPs. So on the left are ATAC-seq peaks that are gained, stable, or lost in GMP when RUNX1 is deleted, and on the right are ATAC-seq peaks in neutrophils. And in both cell types, more ATAC-seq peaks were gained than lost. You can see that in the heat map and also in these peak profile plots below the heat maps. And this indicates that RUNX1 loss in GMPs is increasing the genome-wide accessibility in both GMPs and neutrophils. Many of the Go biological terms associated with peaks that were gained in neutrophils when RUNX1 was deleted are related to immune responses, and this correlated with the increased inflammatory cytokine production we observed from these cells. Similar Go terms were enriched in peaks that were gained in GMPs, indicating that this enhanced immune response program was established in the GMPs. Now, to address the question of whether the chromatin changes related to immune responses in neutrophils originated in GMPs, we examined whether the peaks gained in RUNX1 deficient neutrophils had also been gained in GMPs. And of the greater than 5,500 peaks gained in RUNX1 deficient neutrophils, 75% of them had been gained in GMPs. So therefore, the chromatin changes that result in an elevated innate immune response by neutrophils originate in GMPs and are transmitted to the neutrophils. Now, genes associated with gained ATAC-seq peaks included several that encoded proteins in the toll-like receptor signaling pathway. So this diagram here on the right shows in green the genes whose transcripts were increased in RUNX1 deficient neutrophils or GMPs based on RNA-seq. On the top left here are sequencing tracks for the CD14 and the TLR4 genes, where you can see that the ATAC-seq peaks are higher in RUNX1 deficient, for example, neutrophils compared to control cells. We had previously shown that TLR4 protein levels were slightly elevated on the cell surface of neutrophils, and here we show that CD14 messenger RNA is clearly increased, and in addition, there's a higher percentage of neutrophils that are CD14 positive. Adding a CD14 blocking antibody to cultures of neutrophils stimulated by LPS decreased the production of inflammatory cytokines to baseline levels, suggesting that this elevation of CD14 signaling could be is contributing to the hyperactive LPS response, and that by blocking CD14 could be a strategy for reining in this exuberant TLR4 signaling in RUNX1 mutant neutrophils. Now, I think we obtained our first real insight into the mechanism underlying the increased inflammation by determining what transcription factors were opening the chromatin when RUNX1 is lost. And we did this by identifying transcription factor footprints. So basically, footprinting is a computational method whereby you can infer which transcription factors are bound to DNA, because the enzyme that's used to cleave DNA cannot cleave where a transcription factor is bound. And this results in a dip in reads in the middle of an ataxic peak from which you can infer which transcription factor is bound based on the conservation of the motif under this peak. So we performed a bias-free footprint enrichment test to determine which transcription footprints were enriched in peaks that were gained or lost when we delete RUNX1 in GMPs. So these are the peaks that are lost, and as you would expect, when we delete RUNX1 in GMPs, the most highly enriched transcription factor footprints that are lost were for RUNX1 itself, and also of GATA proteins in both in GMPs. But more interesting for the perspective of this story was that the most highly enriched footprints and peaks that were gained when RUNX1 was deleted that are conferring this innate immune program were those for multiple interferon regulatory factors and STAT1, STAT2. And not only were the footprints for the IRFs and STAT1, STAT2 enriched in the gained ataxic peaks, but the IRFs and STAT1, STAT2 appear to be opening the chromatin around them. So these, the footprints in these plots are centered on the STAT1, 2 and the IRF motifs. The x-axis is base pairs and includes 100 base pairs on either side of the footprint. And the y-axis is normalized read counts, which reflects the accessibility of chromatin. So these gray lines represent the average read counts for the control cells, and the teal lines represent the RUNX1 mutant cells. And you can see that the chromatin around the STAT1, STAT2, and the IRF2 here footprints gain accessibility significantly in neutrophils, indicating that not only are these transcription factors binding to these sites, but they're opening the chromatin in their local vicinity. And there was also a trend towards increased opening of the chromatin around the STAT1, STAT2, and IRF footprints in GMPs. STAT1 dimer footprints, on the other hand, were not enriched, and the chromatin around them was not more accessible. And so the enrichment was specific for these STAT1, STAT2 heterodimers. So the IRFs, STAT1 and STAT2 are downstream transcription factor effectors of type 1 interferon signaling. So the fact that the footprints for these transcription factors are enriched in the open chromatin indicates that this signaling pathway is more active in RUNX1 deficient GMPs and neutrophils. As I mentioned, STAT1 dimer footprints were not enriched, and STAT1 dimers mediate or the transcription factor effectors of type 2 interferon signaling. And since they were not enriched, this means that really the predominant pathway that was activated was the type 1 interferon signaling pathway. And this state of elevated expression of a type 1 interferon signaling and type 1 interferon stimulated genes is referred to as a type 1 interferon signature. And type 1 interferon signatures are typically found in autoimmune diseases such as lupus and rheumatoid arthritis. Now, there's several possible mechanisms for the presence of an interferon signature. And one possibility is there's just increased levels of type 1 interferon, and that would activate this canonical signaling pathway, which involves dimerization of the receptors, activation of the JAK1 and TYK2 kinases, phosphorylation of the STAT1, STAT2, this complex that these proteins form with IRF, and then translocation to the nucleus and the activation of interferon signaling genes. However, we were not able to detect elevated levels of type 1 interferon anywhere in the blood, in the bone marrow, or from neutrophils purified from RUNX1 mutant mice. And this really cast a doubt on the notion that this elevated type 1 interferon signature was generated as a result of increased levels of the ligand type 1 interferon. But another way that cells could acquire an interferon signature is through the increased activity of a non-canonical interferon signaling pathway. So, for example, basal interferon signaling can occur in the absence of interferon and without dimerization of the receptor or the activation of JAK1 and TYK2 by a non-canonical pathway in which unphosphorylated STAT1 and STAT2 form a complex with IRF and then activate transcription. And the way the non-canonical pathway could be activated is by increasing the levels of the IRFs and the STAT proteins themselves. And in support of this model, we detected elevated levels of transcripts for STAT1 and for a number of the IRFs, and increased levels of STAT1 protein in Western blot in RUNX1 deficient neutrophils, about a twofold increase in STAT1 protein levels. We tested then whether the interferon signature in RUNX1 deficient neutrophils was the result of elevated canonical or non-canonical signaling. So shown here is the basal increase in the basal expression of 3 of the interferon stimulated genes, STAT1, GBP2, and IRGM2. And so to test our hypothesis that the elevated expression of these genes in the absence of interferon, so this is basal expression, was not caused by elevated levels of interferon in vivo, we treated neutrophils with 2 inhibitors of the canonical signaling pathway, an antibody that blocks the interferon receptor and ruxolitinib, which blocks the JAK kinase. And we determined whether these inhibitors affected the basal expression of interferon sensitive genes. So the rationale for this experiment is if the interferon signature is due to increased signaling through this canonical pathway mediated by increased levels of type 1 interferon that we were just unable to detect, then either the blocking antibody or ruxolitinib should return the expression of interferon sensitive genes to baseline levels. However, if the interferon signature is due to elevated non-canonical signaling, then neither the blocking antibody nor ruxolitinib should affect the basal expression of interferon-sensitive genes. And here are the results. So the data showed that the canonical pathway, which we intentionally activate by adding type 1 interferon to purify neutrophils ex vivo, is robustly inhibited by the receptor blocking antibody, as evidenced by the decreased expression of these 3 interferon sensitive genes. And this has really served as a positive control for the experiment. However, the basal expression of interferon sensitive genes is not inhibited either by the receptor blocking antibody or by ruxolitinib. And that indicates that these increased levels of this interferon signature is due to increased activity of a non-canonical signaling pathway, and we think this is because of increased levels of STAT and IRF transcription factors. So here's a summary of what I've told you so far. So RUNX1 either directly or indirectly restrains non-canonical type 1 interferon signaling in both GMPs and neutrophils, and also RUNX1 also directly or indirectly represses TLR4 signaling in response to LPS in neutrophils. And it does this at least in part by de-repressing the expression of the co-receptor to TLR4, which is CD14. And this results in an overproduction of inflammatory cytokines and chemokines in response to LPS. To gain insight into whether RUNX1 is directly or indirectly restraining non-canonical type 1 interferon signaling or TLR4 signaling, Daniel Yen, a new MD PhD student in the lab, and Alex mapped RUNX1 occupancy in the genome by cut and run to determine if RUNX1 binds to genes encoding proteins in these pathways. And they did this in GMPs because that's where the important genetic changes are occurring when RUNX1 is lost. So we found that RUNX1 directly occupies multiple genes in the type 1 interferon signaling pathway. And here's an example of RUNX1 occupancy of the IRF1 gene. There are 5 RUNX1 bound sites in IRF1. You can see, these are the ATAC-seq peaks. You can see that there's a gain in the ATAC signal in RUNX1 deficient GMPs. And one of these gain peaks corresponds to a RUNX1 bound site. And in fact, when we compared the chromatin accessibility of all the genes encoding proteins in the type 1 core signaling pathway, we found all these genes in the pathway listed here are directly occupied by RUNX1, and all of them had gained chromatin accessibility in GMPs, and most of them gained chromatin accessibility in neutrophils when RUNX1 was deleted. And we found the same is true for the TLR4 pathway. All but 2 of the genes listed here encoding molecules in the TLR4 pathway are occupied by RUNX1, and the accessibility of all of them increases in GMPs and most increase in neutrophils when RUNX1 is deleted. So this refines our model. We think that RUNX1 directly binds to and may directly constrain the chromatin accessibility of multiple genes encoding proteins in both the type 1 interferon signaling and TLR4 signaling pathways. We then started to look at this potential interplay between type 1 interferon signaling and toll-like receptor signaling. And the approach we took was to block one pathway and assess its effect on the activity of the other pathway. So for example, to determine whether elevated type 1 interferon signaling increases the TLR4 response, Alex stimulated control and RUNX1 mutant neutrophils with LPS in the absence or presence of the JAK inhibitor ruxolitinib. And the reasoning is that if elevated type 1 interferon signaling is augmenting the TLR4 signaling response to LPS, ruxolitinib should block that effect. And indeed, Alex found that ruxolitinib could ameliorate this hyperinflammatory neutrophil response to LPS, demonstrating that elevated type 1 interferon signaling in RUNX1 deficient neutrophils is amplifying toll-like receptor 4 signaling. To do it the opposite way, determine if increased basal TLR4 signaling was contributing to the interferon signature, Alex examined whether deletion of the TLR4 co-receptor CD14 expressed the basal expression of interferon-sensitive genes. So Alex isolated neutrophils from mice in which RUNX1 was either deleted in GMPs and mice in which RUNX1 was deleted in GMPs, and these mice also had a germline deletion of CD14, and found that loss of CD14 did not decrease the expression of interferon-sensitive genes, and therefore the elevated type 1 interferon signature is not caused by increased basal TLR4 signaling. So therefore, these data demonstrate that the interferon signaling sensitizes the toll-like receptor signaling pathway, but increased basal toll-like receptor signaling is not responsible for the type 1 interferon signature. So interferon signatures have been associated with the increased chromatin accessibility of retroelements. Retroelements are repeated sequences that constitute about 42% of our genome. And there are 3 main classes of them. There's the LTR retrotransposons, the LTR ERVs. There are long interspersed nuclear elements or lines, and short interspersed nuclear elements, the signs. And normally, retroelement expression is held in check in differentiated cells through epigenetic mechanisms, but retroelements can become activated in inflammatory and autoimmune diseases. And listed here are a collection of autoimmune and inflammatory diseases where de-repression of retroelements has been seen. So we examined the chromatin accessibility of retroelements in our ATAC-seq data and found an overall increase in gained peaks in both RUNX1 deficient GMPs, and neutrophils. And the gained ataxic peaks were specifically located in line and LTR-ERV retroelements. Interestingly, there were only a small number of transcription factor motifs that were enriched in the gained peaks, and these included the IRFs and STAT2. So these data suggest that the elevation of non-canonical type 1 interferon signaling is directly increasing the chromatin accessibility of retroelements. Alex also determined if the increased chromatin accessibility of retroelements results in their increased expression. So she used an antibody specific for double-stranded RNA, which is produced when retroelements are transcribed, and then analyzed double-stranded RNA levels by flow cytometry. And she observed an increase in the amount of double-stranded RNA in RUNX1 deficient neutrophils, although not in RUNX1 deficient GMPs, also not in RUNX1 deficient monocytes, which are not hyperinflammatory. So we don't yet know whether these elevated levels of double-stranded RNA from retroelements is actually contributing to the hyperinflammatory properties of RUNX1 mutant neutrophils, or if they're just correlate with them. But we're in the process of mutating some of the sensors of cytoplasmic nucleic acids to determine if this double-stranded RNA is simply a consequence of the increased accessibility of the retroelements, or if it also contributes to the inflammatory properties of RUNX1 deficient neutrophils. So one reviewer asked us, given that the de-repression of retroelements is known to occur upon the global de-repression of chromatin, whether there was any global de-repression of chromatin in RUNX1 mutant cells. And the best way to assess this is by doing a chromatin capture analysis to determine whether there was a redistribution of chromatin from the heterochromatic B compartment to the euchromatic, more active A compartment. But we found that the A and the B compartments, for the most part, did not change much in RUNX1 deficient GMPs and neutrophils, and there was a very limited transition between the two departments. So we think that the retroelements are they're primarily located in the A compartment, and that they are primarily being activated directly by type 1 interferon signaling. Retroelements and innate immune system, the innate immune system co-evolved during evolution, and many retroelements are known to function as transcriptional enhancers of innate immune genes. So we hypothesize that this increased accessibility of retroelements in RUNX1 deficient GMPs reflects their increased activity as transcriptional enhancers of the innate immune response genes. So my final of these summary slides is that we believe that RUNX1 binds to and likely constrains the chromatin accessibility of multiple genes in both the type 1 interferon signaling and the TLR4 signaling pathways. This subsequent elevation of non-canonical type 1 interferon signaling further contributes to the hypersensitivity of the TLR4 signaling pathway, for example, by elevating the expression of CD14, which is also an interferon stimulated gene. We think that increased non-canonical type 1 interferon signaling is also contributing to the increased chromatin accessibility of elements. Now, all of these epigenetic changes are happening in GMPs when we knock out RUNX1 in GMPs, and they are inherited by the neutrophils that differentiate from the GMPs. And we propose that this inflammatory state caused by RUNX1 loss is a form of maladaptive trained innate immunity. So trained innate immunity is induced by infection by a pathogen or by chronic inflammation. And both of these inducers of trained innate immunity cause epigenetic rewiring of HSCs and progenitors, including GMPs. So these progenitors and HSCs acquire an innate immune memory, and this innate immune memory is then propagated to differentiated immune effector cells, including neutrophils. Now, innate immune memory that's induced by infection with a pathogen is a beneficial thing because it will result in a more robust response to that pathogen upon a repeat infection. Maladaptive trained innate immunity, on the other hand, can exacerbate systemic inflammatory diseases. So what we're proposing is that RUNX1 loss causes a fixed state of maladaptive innate immunity, which could be contributing to the inflammatory conditions observed in FPD-MM patients, perhaps even including their overly robust response to vaccines, and could also be contributing to an inflammatory bone marrow milieu that accelerates clonal hematopoiesis by selecting for HSCs that have acquired somatic mutations. So I think some interesting and important questions going forward are, do RUNX1 mutations cause fixed maladaptive innate immunity in other cells? And I believe the answer to this will be yes, that this phenomenon will not be restricted to neutrophils. There's anecdotal evidence in the literature. I've heard anecdotal evidence at meetings, and also there's a report in the literature that RUNX1 mutations cause hypersensitivity of B cells. A new graduate student in my lab, Daniel Yen, wants to examine maladaptive innate immune memory in FPD-MM HSCs. Do hyperinflammatory neutrophils or other cells accelerate clonal hematopoiesis and contribute to the increased leukemia risk in FPD-MM patients? And if so, would drugs used to treat patients with autoimmune diseases associated with type 1 interferon signatures reduce the leukemia risk? So I think this causes me anyway to think in sort of different terms about, what is the core problem that is causing this inflammation, and what are the, essential signaling pathways that could be involved? And type 1 interferon, we think, is probably a major one. So finally, these are the people who did the work. This is the Speck lab, FPD-MM team. Alex Zazulin did most of the experiments I showed you today. The project was started by Dana Bellissimo, a former MD-PhD student in the lab, and Daniel Yen, a new MD-PhD student, is going to carry it forward. We had help from Darwen Yi from Andy Min's lab on the double-stranded RNA analysis. We had help from Jingyan Ren in Wei Tong's lab on the Western blots. Wenbao Yu is our extremely essential bioinformatics collaborator, and he's actually a co-senior author on this paper that we're preparing to resubmit. We obtained the CEBP alpha Cree mice from Evo Tau at Erasmus University, the RUNX1 R174Q mice from Lucio Castilla at UMass Worcester. We're collaborating with Paul Yu at the NIH to analyze patient neutrophils. And many thanks to Ellen Rothenberg and her great postdoc Boying Shin for providing to us an optimized cut and run protocol for RUNX1. So thanks very much for your attention, and I'm happy to take any questions.
Thank you, Nancy. What an incredible presentation and such beautiful mechanistic work. We have some questions, and so I'm going to just kick off with wonderful talk, Nancy. I'm fascinated by the data RUNX1 directly represses type 1 interferon pathway. We had similar data from AML patients showing RUNX1 mutated AML has increased type 1 interferon and also PDC, dendritic cells. I'm blanking on what P stands for.
Yeah.
Thank you. Previous studies showed that PDCs are reduced in MXCRE RUNX1 knockout models. In your GMP RUNX1 knockout model, have you looked at plasma dendritic cell differentiation? Thank you.
No. So who is the question from?
Oh, this is from Wenbin Zhao, who is an early career investigator for the RUNX1 research program.
Yeah, Wenbin, I think the neutrophils were only scratching the surface a bit of what's actually being, what cells are actually producing these inflammatory cytokines. So if we stimulate whole bone marrow and look at what inflammatory cytokines are produced, there's many more than the few that we find overproduced by RUNX1 deficient neutrophils. So yes, I think there are other cellular sources of inflammatory cytokines being produced. Other sources of cells that are producing these inflammatory cytokines. There could also be crosstalk between these cells in the bone marrow, where one is stimulating another. And yeah, dendritic cells is definitely a place that a cell that we should be looking at more carefully. I agree.
I just have a follow-up question, actually. With interferon pathway signaling, are there specific cytokines that are associated with that, pro-inflammatory cytokines associated with that pathway, or is it a very large spectrum of cytokines?
It's pretty, I think there's about 150 ISGs, interferon stimulated genes, so it's going to be fairly
robust.
Broad. And then if you think that interferon signaling is also sensitizing other signaling pathways, it's a sort of scary thought, right? It's having very, very broad effects, I think.
Right. So we have a question. We have multiple questions from Dr. Tim Trish. And so I'm going to kind of read them in a row because it looks like they're all slightly linked. So what do you suppose is the mechanism for transmitting open chromatin features from progenitors to progeny over mitosis? And then that's followed up with, also, at what level was the high C analysis done at? Single cell or bulk in GMPs or neutrophils or a mix of cell types? Is it possible? Should we stop there and then we'll continue?
Yeah. Let me answer the second question first, because I have a horrible memory for a chain of questions. So we did it, the high C was done in GMPs and in neutrophils, and it was done by bulk sequencing.
Great. Is it possible that the heterochromatic derepression is more of a variability thing than a fixed effect in RUNX1 FPD?
I think it has to do, I think a lot of it's coming from these retro elements, to be honest, because there's so many of them. And I think it's kind of related to the type 1 interferon signaling pathway and the activity of the transcription factors downstream of them. And the fact that we find, this collection of a pretty small number of motifs that are really highly enriched in those retro elements, I think it suggests that it's not a random thing, but it's actually a kind of a directed phenomena.
And then he asks a follow-up question. Is there any sex bias in RUNX1 FPD or in transformation to clonal hematopoiesis, MDS, and AML?
I am not aware. Maybe Wenbin can answer a question about MDS and AML.
I don't know how to allow Wenbin to speak, but I think I do recall from Dr. Liu's data, the recent bioRxiv paper from the natural history study that he and his team are conducting. I believe there was a bias, or maybe it's from the retrospective RUNX1 database from Anna Brown and Hamish Scott in that group. So forgive me if I'm forgetting the source of this, but I do believe that there is a sex bias where there is a higher a bias for females in AML.
Wenbin is here, if you'd like to.
Oh, great. Go ahead, Wenbin, if you want to speak to that question.
Hello, can you hear me?
Yes,
Yep.
we can.
Yeah, I'm not aware of that, but the new data from Paul Liu, I haven't read extensively yet. I downloaded, but I had no time to read yet. I mean, somatic RUNX1 mutation has no sex bias. That's for sure.
Mm-hmm. Thank you. Appreciate it. Okay, so I will follow up with that question because I'm fairly certain I saw a sex bias in one of those cohorts, whether it's the retrospective data set in the RUNX1 database or whether it's in the natural history study. So I'll follow up. A question from Connie Bonifer. Hi, Nancy, lovely story. Very similar to what we saw in Hodgkin lymphoma. Question, GMPs do not just give rise to neutrophils, but also macrophages. And I know CD14 is really important in macrophage as well. Do these cells not show an epigenetic imprint? Why do you think that is? Macrophage memory involves AP1 complexes. Is that an override?
We did not look. So we did not actually look at macrophages. We looked at monocytes and we did not see increased production of inflammatory cytokines in monocytes. So we did not study them. I couldn't convince Alex to study a cell that didn't have a phenotype. So, but I think it would be a really interesting comparison, but probably should be done in macrophages, I'm thinking.
Any insights as to what is happening in humans and in RUNX1 FPD patients, and whether you're seeing any of these effects in the neutrophil compartment or the GMPs at this point?
Yeah, we have been able to successfully do ATAC-seq on RUNX1 deficient on FPD neutrophils. I forgot to, did I acknowledge? I did acknowledge Paul Liu. So he's very kindly opened up his lab to us. And when he enrolls a family who comes with a family member, we travel to the NIH and get a sample of blood because neutrophils are so easily activated. We want to process them side by side. So we analyzed neutrophils from a child and from a 40-something-year-old. And the child had no evidence of an activated neutrophil phenotype, but the adult had a really evident neutrophil activation phenotype. So I don't know exactly what that means. You know, the disease is, leukemia is partially penetrant. The inflammatory disorders are partially penetrant. There's a big age difference there. There was a difference in the mutations. I think we're going to have to analyze a lot more samples before we can say anything definitively. That's a lot more trips to the NIH.
Great, thank you. And is there any, do you have any hypotheses as to whether any of the common co-occurring somatic mutations that occur in patients could somehow be linked to the sort of maladaptive innate immunity that you see? Is there any sort of connection to some of the common somatic mutations that we see and this process that you've uncovered in the neutrophil compartment?
Yeah, well, so since B-core is so common, and it's also common in aplastic anemia, which obviously is also immune disorder, I think there must be some fitness that B-core is providing to a RUNX1 mutant HSC that's allowing it to preferentially expand. With respect to other mutations that have been seen in clonal hematopoiesis, you know, I don't know. I think it's an interesting question if as you continue to accumulate mutations, you increase the inflammatory properties of the downstream myeloid cells. I had a really interesting talk from John Dick at a stem cell meeting last week where he had identified this two different HSC compartments, one that has a very kind of memory-like inflammatory signature. And in patients who had COVID, that compartment had manifested big changes. And in patients who had clonal hematopoiesis, that compartment also changed in the normal HSCs in those patients. And so it seems like the inflammatory milieu is not just affecting it's affecting all HSCs in the bone marrow, even though you have only a clone of these cells producing inflammatory cytokines.
That's fascinating. Well, I can't believe it, but we have, oh, I just see another question. Let's see. Oh, not a question. This was a phenomenal talk. Thank you. And Dr. Zhao and Erickson for your patience, incredible concentration of expertise. Wow, very, very kind comment. Thank you very much.
Thank you very much.
Thank you everyone for attending. And if you have any further questions, just you can send them directly to me and I'm happy to follow up with Dr. Speck. And again, certainly appreciate your attention and very much appreciate your work, Nancy. It's moving us forward, that's for sure.
Oh, thanks very much. My pleasure.
Take care. Thanks, everyone.
Bye.